Good afternoon, everyone, and thank you so much for being here with us on this very sunny afternoon. And I'm super pleased to introduce this March webinar of Invasive Climate Change Education Solutions Network. I'm here with my colleague, Professor Janet Vale, and we'll be co-moderating and co-hosting this webinar together. My name is Elena Lubimtseva, and I have a great privilege to introduce today our guest speaker, Dr. Trisha Smrecak, who is the education coordinator at Inland Seas, a nonprofit organization located in the Suttons Bay in Michigan. Trisha is not new to GVSU. She previously was a professor of geology here. She's a paleontologist. She holds a PhD in geology from Michigan State University, and she is also an adjunct professor at Northwestern Community College. She previously taught in several other institutions. She's an expert in climate change education, environmental education, aquatic education, and I'm very, very excited to welcome her today to this miracle webinar. So thank you, Trisha, for joining us. Welcome, and I am just so grateful for your presence today on the CCSN webinar.
Thanks, Elena. Thank you. And thanks, Janet. It's, I don't know about expert, we're all learners, right? So I just prefer to do my favorite learning outside in the environment and frequently on the water. But so yeah, I'm here today to share with you some of the really interesting changes that we've observed on Lake Michigan. As Elena said, I work at Inland Seas Education Association, and I'll tell you all about what it is that we do and who we are. But the spoiler alert is that we've been doing citizen science since before it was the cool thing to do. And we've got a really cool track record of data. And so if you were here last month or you've watched the videos, you saw another program that does many of the same data collections or similar styles of data collection. So you'll hear a little bit about where our skills and programs overlap and where they're different. And also I wanted to focus on some of the data that we got so that you guys learned a little bit about the methods, and so we can see what these methods have been showing us about Lake Michigan. So that's where we'll be talking today. There we go. I just highlighted a state of the lake report here. I think it's really important for us all to consider how lucky we are to live in this state. We have abundant resources, but we don't realize how lucky we are, how threatened those can be. And even within the Great Lakes, you know, the fact that in the last few years, we've had people who get their drinking water from a Great Lake not be able to use the drinking water in their house. This has become, you know, more and more systemic, and we have abundant resources. So it's thinking about stewarding, being stewards of these Great Lakes resources is so important. In the context of we're stewards of the largest supply of this nation's water, and one of the largest supplies of surface freshwater on the planet. So, so it's just really good to put some context in. The little corner of the Great Lakes that we steward or call home is Suttons Bay. It's right here. Our primary education vessel is the pretty sailboat you see there with bright red sails. That is the sailing vessel Inland Seas. She's a 78-foot schooner. And we take folks out on that schooner to do sampling. So our sampling is a little less precise, and we have far fewer technological pieces of equipment in the state of things that have batteries and screens, and we have a lot of mechanical advantage pulleys and those sorts of technologies aboard our vessel. So that's a little bit about the place that we educate from. We take that ship. Our primary home is in Grand Traverse Bay here, which is a part of Lake Michigan, and in the little inlet here of Sutton's Bay. But we travel for the last 3 years. We've been in 4 out of the 5 Great Lakes, I believe. And Lake Ontario keeps getting missed, but we'll get out there one of these days. We also recently bought another vessel, the Alliance, which will be coming home to Sutton's Bay through Lake Ontario. So we're going to get there this year from Virginia this spring. So if you're interested in hearing about a voyage like that, keep track of us on social media, because we'll be sort of sharing tidbits of that. And I think it's going to be a fun way. A lot of our teachers are planning on sort of zooming in on where the inland seas or where the Alliance is going to be. They're following along. Anyway, so our mission is to inspire a lifetime of Great Lakes curiosity, stewardship, and passion in people of all ages. And here's some pictures. We are all about the experiential component of authentic, using scientific equipment to collect samples and then analyzing them. So you can see we've got people holding fish that are collected with an otter trawl here. We take a 10 minutes sample every single trip. We take the same sample. And so what you'll hear a little bit about is that we've got a longitudinal data set that shows us what happens when you go fishing for 10 minutes in the same place over 30 years, what you catch. But in the moment, in the 3 hours you spend with us, you're immersed in touching, holding, measuring those fish, or searching for macroinvertebrates on plant life, or in the benthos, the bottom of the Great Lakes. We take a scoop of that, or examining plankton under a microscope. There's another picture of mud. So if you don't get plant life, you get mud life. And so these are just ways that we bring the Great Lakes to life and the different parts of the Great Lakes food web to life. And so when someone steps foot on our ship, we say that they are going to be 3 things. And we say this whether you're a 4th grader, a 12th grader, or a 27th grader. We say that you're going to be sailors, scientists, and stewards, because those are the things that matter a lot to us. And so you act as a scientist for the day. You are the ones who are collecting those plankton samples, who are dropping the Secchi disc. You are the ones helping to throw our fish net, our otter trawl, over the side of the ship, and recording the latitude and longitude. So you are doing that work. And then you were a sailor, you help us raise the sails. You help coil the lines, you help bring up the anchor, you do all of the things that sailors need to do to successfully manage a vessel. And we hope when you leave that, because of the connection, the emotional experience you've had, which we hope is a good one, you leave caring a little more, knowing a little more about the Great Lakes, and wanting to be a steward of that space. One of the best ways to be a steward, because we aren't all lucky enough to live right on the water, is that connection to watershed, right? I think a recent study, and I can't, said something like upwards of 60% of people can't define what a watershed is. It's and we all live in one, or we can say we know we live in one, but what it actually is. And watershed stewardship is the most important way we can protect the Great Lakes. And the Great Lakes are experiencing a lot of pressure from a lot of different angles, for sure. And so, and we'll get into what our data says about that in a couple of minutes. But I think it's important to show someone that protecting their own watershed is the best way to steward the Great Lakes. So we have in the last few years expanded onto on land programs into our watershed as an example of ways that we can learn about and monitor our watershed so that we can be better stewards of the Great Lakes. We do that through things like macroinvertebrate collection, which maybe you're familiar with, and water quality, water chemistry tests of streams, but we also take transects of the riparian zone. So we look at the area surrounding a water body immediately adjacent to a stream or a coastline, and we check it out. How resilient is it to flooding? How is it contributing to filtering pollutants and other things? How is it acting as a nursery for our native fish and plants? And so we explore some of those things as well. We also really think it's important to focus and highlight the careers that are here on the Great Lakes, because the next generation of stewards are also our next generation of sailors, of freighter captains, of construction workers that are building homes and seawalls and things on the Great Lakes. And so choices that we make in our careers can also be really impactful, and we can impact in positive and negative ways. So we talk about things like navigation. We have a navigation program that teaches you all the math necessary to navigate, and how to use a compass and chart a course, and how to use an ROV to operate, to find sunken ships, and what the Coast Guard does with sunken ships if they get found. And so talking through some of those activities. So those are other ways that we connect kids to, and anybody to the Great Lakes. Through the work that we've done, we've been around for over 30 years, and every time a class signs up to go out with us, we thank them because you signed up, we get to go out and take another sample. And so for over 30 years, we have gone out from when the boat can get out on the water to the, early in May to the second to last, or sometimes last week in October. And we take folks out, and when we go out, we take a set of samples. And so twice a day for many of the days between May and October, we have data on the fish populations, on the plankton populations, on what's happening at the bottom, the benthos of the Great Lakes. You see a Secchi disc, which if you were here last month, you heard about as a way to, we'll talk about what that is in just a minute, but looking at clarity, exploring plankton, the light levels, how far down the light goes. We also take air temperature, we take water temperature at the surface, we take water temperature at the bottom, and when I say we, I mean the students, the participants, whoever's out on a trip is taking that data. So I'm going to highlight some of what our data shows for us today. Yeah. So I think, what does it mean to monitor? I think that's a great analogy question I like to start, and sometimes we'll talk about this, especially with younger groups on the ship. If you think of your favorite movie and you freeze frame it, what are you learning about your favorite movie? If you show me your favorite movie, what can I know about it in one frame, right? In one frame of a football game, is this a kickoff return? Is it the game-winning extra point? What's going on in that frame? In this frame over here with poor old Charlie Brown, what happened with the football there, right? So one moment doesn't tell the whole story. It just tells one thing. And there's a lot we can get from that. but it doesn't tell us everything. And so I like to think about monitoring efforts or longitudinal studies as watching the full movie or getting a chance to read the full story. And so every single sail is one frame, right? Is Charlie Brown laying on the grass in shame, knowing he shouldn't have tried to kick the ball again. But over 30 years, each class that's contributed data helps us to paint the picture or tell a story of the health of the Great Lakes. And so what you see on the screen here is a Secchi disc. I also wore my Secchi disc earrings for the occasion. And a Secchi disc is light and dark colored. It's black and white, and that's just, it's high contrast. I have a 4-month-old at home. And so I know that that's the first thing a baby can see, right? That's why books are in black and white for little babies, so they can see high contrast. It's really easy for our eyes to pick up. And so that high contrast object gets dropped into the water. We do this at 5 different locations on our ship, and we take the average data. We drop it down and we see how far down we can see that Secchi disc, right? And that is going to tell us how clear the water is. And it tells us something about how far the sunlight penetrates. But going back to that analogy, what can one single measurement of clarity tell us? Probably not a lot. But if you take a measure every day for, say, the whole sailing season, then you can get at a story. So this is 2018, and our x-axis is May through October. So we aren't out any earlier than May, and we put the boat to bed in October. And ice, not this year, but most years, covers the bay for a good portion of that. So we wouldn't be able to get a Secchi depth another way, but we have these summer months. And so what we see is the average monthly Secchi depth in Sutton's Bay. The y-axis is depth in meters. I think you probably all can notice that it starts out, you can see pretty far down in May and June, but by the end of summer into September, it's almost half the depth, going from 16 meters to, well, sometimes it's 8, I guess that's 10, so less than half, but you can see a lot less far down. The clarity decreases. And so what does that mean? As a person who likes to go swimming, maybe you love clear water, so you prefer swimming in June. But as a fish or a native clam, that might say something else about your preferences, right? And this is one season. What is it doing over multiple years? And it asks, a scientist then asks the question, why is it doing that, right? And so we tell that story through the data that we collect, I guess is how I'll say that. Before we put the monthly to bed, I want to show you another interesting trend, because we can look at this data not just for 1 year, but for 30 something years. This is our monthly averages in Secchi depth from 1995 to 2005. All right. So you can see the trend is a little less obvious, but you can see that early in the season, you can see a little more deeply. By the end of the season, at the end of summer, you can see a little less deeply, right? The clarity decreases. You also might notice these little whisker bars. That's what the data over the years is showing us, the variability, right? And so I want to pull back to 2018 and notice this 1 year we were at around 17 meters in May and June. the average from 1995 to 2005 is about 11 meters. In September, it's about 10. All right. So interesting. Now, we're going to look at the same monthly averages whisker chart, but instead of 95 to 2005, we're going to look from 2010 to 2020. Something's happening. Something interesting is happening. That trend of being able to see really, really far down early in the season is a lot more obvious in more recent years. And so scientists ask why. And because we say when you're on the ship, you're a scientist for the day, we ask why, and we ask the students why. And so today we're going to ask why. All right. And so we need to zoom out. Instead of that seasonal trend, let's look at what's happening from 1995 to the last few years. So this one is 1995 to 2019. Now, annual, so average Secchi depth over the whole year, we take all those numbers from a year, we lump them down to one data point, taking an average. In 1995, the water was a lot less clear than it was in 2019, right? This past year, we are still chugging the numbers for, but 21, I think it was almost 16 meters was the clarity. So it is getting clearer and clearer, which is great if you like swimming in Sutton's Bay. But what makes the water clear? What makes the water change clarity? Why in the last 30 years has it gotten clearer? And why does it get less clear from the beginning of summer to the end of summer? And when you ask a kid on a sailboat these questions, a lot of cool ideas come through. Things like, well, people go swimming more in the summer. The boats are churning them up. They're making really great hypotheses. And so as an educator, I'm going to sit there and listen. Yeah, that's a great. How would we investigate it, right? And so we talk through some of those options. And then a lot of times what we'll say is, I think you're going to see something on the ship that you collected today that's going to answer that question. So, oh, I'm sorry. One of the other, I wanted to throw this case study in because one kid really went far with this. I've heard it a few times, but a kid said, well, the water's getting deep, right? Like they knew about how high the water levels in Lake Michigan had been. And so they said, well, it's probably getting clear because the water's getting deeper. I said, well, that's a really cool hypothesis and we can look that up. So here are Lake Michigan water levels. Pull us over this way. For the last about 100 years. And yeah, it has gotten deeper. It's getting a little, it's getting closer to average again this past year. But since our data set started around 95, which is right there, well, it was deep. It was deeper than average and then got shallower than average and then got deeper than average, but we didn't see that trend, right? So we can talk that through. But it's a really cool observation. And the fact is that kids getting this data and starting to like piece apart this longitudinal data, they start asking those questions too, which I think is just so fun because the only thing that separates me as a scientist from them as a scientist is a extra background knowledge and a little more time and experience. And so we can look at that. We can talk about rolling averages. I'm going to fast forward in my brain. Some kids will say, well, maybe it's raining more. I heard climate change means it's raining more. Oh, okay. Well, that's a great question. So, or a great hypothesis. Let's compare the data. And so I do want to point out that it's interesting because if you look at, this is all 5 of the Great Lakes, which scientists consider Michigan and Lake Huron the same lake. So that's the green checks for you, but this is all the Great Lakes, and you see that they vary, their water depth varies. But by and large, especially since about 1970 or so, and we can look at it either with this finer resolution graph from about 1970 or so, 1965, the amount of time the Great Lakes have been higher than average water level has exceeded the amount of time it has been lower than average waterfall or water levels. And that means that there's more erosion happening. So again, when you look at all 5 of the Great Lakes, that trend continues, right? So all 5 of the Great Lakes are having this common experience. A lot of folks talk about climate change and how much more precipitation we've been getting, and that's a really great observation. So that's we can pull that data in too. Now we collect a lot of these pieces of data, but when you put our data set against some of these bigger data sets, you really start seeing a cool picture. Great Lakes Basin precipitation since 1965. This is the average from, I think, 1901 to 2000, the average rainfall that the basin receives. You can see that since about 1968 or 69, we've gotten higher than average rainfall every single year. Precipitation could be snowfall as well. And there's a lot of, this is tied intimately to climate change. The interesting thing is, that's not necessarily, that's not what we're seeing when we're looking at Secchi depth. So that's a lot of times what our kids are thinking, but it's a really cool story. We're going to get back to climate change, but now I have to zoom into the bugs and the creatures, because that's when we're monitoring at our basin level, at our little sub-basin, we're seeing more of the changes in the animals, and the changes in the animals have impacts on the changes in the lake, or are feeling those same impacts from climate. So pulling back to monitoring monitoring our Secchi depth, when we measure Secchi depth, remember we asked the kids why that might be changing. We need to think about what sunlight is controlling. So Secchi depths actually, in a lake system, in a river system, it's frequently sediment or dirt or things that are eroding, clouding the water and making it hard to see. In a lake system, that stuff settles to the bottom really quickly. And instead, the things that make the water cloudy is life, is the creatures, these single-celled and very small multicellular organisms that that make up the base of the food chain, the plankton that we catch. And so Secchi depth becomes a proxy for how much plankton, how much food is in the lake. And then when we look back at that scale of across one season, why would we see less, why would we have more water clarity or less plankton in May than we do in September and October? And kids, the light goes off. Oh, well, reproduction, they're making more, there's more leaf cover, there's more food, and they start making that connection. And then you say, well, okay, doesn't that happen the same way every year? And the students say, yeah. So why is it changing over time? Over the last 30 years, why is Lake Michigan, why is Sutton's Bay getting clearer? And then the light bulb really goes off, right? And to do that, to find the answer to that, we go to another creature that we collect in our Great Lakes sample, right? When we take a benthos sample using this cute little ponar guy, I call it the claw, we pull up quagga mussels. Now, a lot of folks have heard of zebra mussels. And we used to pull up zebra mussels too, and we still do from time to time, but this is what our sample looks like. Sometimes it looks like this and there are no little clams in there, and sometimes we pull them up and it is just chock-a-block full of clams. And so we are one location, but an interesting thing is that over lots of locations, right? So we've got this longitudinal data set of one place, and then other scientists can take a data set over lots of places, and we'll call that a spatial data set. And so we can look at our place and see how that trend happens, and compare it to other places. And so we caught our first zebra mussels in our first samples. They weren't especially abundant, but we did find them. There's records in our very first samples. We also collected up in Northport, which is way up there at the tippy tip, and in our little embayment here. We've always found them, but they started decreasing in number and we stopped seeing them. But we didn't stop seeing creatures in general. In fact, we saw far more clams in our samples later in time, but they weren't zebra mussels. We also started seeing quagga mussels. And so everybody's heard of zebra mussels. They're common in inland lakes. But quagga mussels are a relative, and they are a little bit better adapted to cold water. So when you look at this pattern, see the zebra mussels are around all the shallow centers. They're frequently around urban centers where there's a lot of warm water drainage pipes. They get in there, they make a real mess of because they like to live on surfaces and they really like that warm water, and the moving water gives them, they've got a food source. They just get really happy in those spaces. But they became far less abundant. They were outcompeted by another invasive clam called the quagga mussel that can handle cooler water, deeper water. And so instead of just in some of those like urban centers and along the shallow parts of Lake Michigan, the quagga mussel, just like it took over our samples in Sutton's Bay, it took over everybody's samples everywhere. And you can actually see an X for where this group sampled in our bay as well. But you can see that they're taking over everywhere in the lake, right? They are the deepest samples we're collecting. Folks are finding quagga mussels in, and we've taken some pretty deep ponar samples. We almost always pull quagga mussels out now. So the loss of one invasive was just replaced by another invasive, and our data bears that out. We can also look into density, and we also saw an increase in the number, a decrease in the zebras, an increase in the quaggas, and in general, an increase in the number of clams. So the quagga mussels have taken over the habitat, and they're also far more abundant. So they're just eating more things. And because a Secchi depth is a proxy for how much food is in the base of the food chain or food web, how much food is in our water, we can actually compare who's doing the eating. Now, these quagga mussels are filter feeders. And so let's see, let's come back here. 1994, 1995, we've got a few zebra mussels. 2000, we start to get a fair number of zebra mussels. By 2005, though, the quagga mussels have taken over. So we've got 2000 and 2005 as data points. So let's look at 2000. From 1995 to 2000, the zebra mussels were becoming established in Lake Michigan. And as a result, they're filter feeding organisms. Their primary food source is plankton. And we see a increase in water clarity because those creatures were eating more plankton year round. They're just there, they're eating. And then by 2010, let's go back here, 2005, the quaggas start taking over. By 2010, they're pretty dominant. By 2005, you can see that water clarity has increased from about 11.5 meters deep to 13 meters deep. And by 2010, we're solidly at 14 to 15 meters water clarity. And we're a little embayment, but that's reflective. The patterns, I'm certain that the patterns, Janet, you see down in Muskegon are the same or very similar. And it's happening all throughout the Great Lakes. It's also interesting because we collect plankton samples, and we don't do plankton density samples, but we do another proxy for plankton density, and we're seeing far fewer species and far fewer, just numerically in an individual drop of plankton, of water with plankton in it from our nets than we have in the past. That data is a lot muddier, so it's a lot harder to present in a simple graph, but we're seeing it. The diversity of the plankton seems to go down. It's tricky. That also changes throughout the season, but it's seeming to go down, and there's just less of it in a drop. And that all is in alignment. So the Secchi depth, the very first sample that we take, and it's so simple because it's just a piece of wood dropped over the side of a boat, tells us a really interesting story once we start putting together those other biosphere components, right? So we're going to crawl up the food web a little bit because the phytoplankton and the zooplankton get eaten by the benthos, right? The quagga mussels. And then they also get eaten by the fish. And that's commonly what everybody interacts with, because rarely do we hold up a cup of Lake Michigan water and go, oh, look at the plankton, right? But we get excited when we catch a fish. So that's the one, that's the part of the food web we're all a little more familiar with. So what are the fish doing? We collect fish in an Otter Trail, which is something, it's a net. These kids are deploying it here, and it's a net that is about 10 feet long, has a little end here that we tie, so the fish swim in this way. It drags along the bottom in the shallow zone. So we're usually about 20 feet deep when we are trawling, and we trawl for about 10 minutes. We try to vary the location a little bit within our bay because it is destructive to the habitat. So we try to every season change our location just a little bit, but we record that, and we are always in the bay when we're taking the sample. So it's a pretty fair snapshot of we're going in the same bay at the same depth, and we're collecting for the same amount of time. So you've got this sweet little girl holding some of the catch, but what's interesting is who we've been catching. Now, this is 1998, this is 2005, and this is a pie chart that isn't showing a number of fish, but instead diversity of our catch. And each of the colors represents a different organism, right? So the yellow here is the three-spine stickleback, for instance. What I want you to pay attention to is this little blue sliver here in 2005. 2005, that blue color is the round goby. The first time we caught the round goby in our nets was in 2005. Here's a picture. They're quite cute. A round goby on some cute little Petoskey stones. And that little blue sliver represents the first time we caught them in our net. So I'm going to show you now 2006 to present. In 2006, the portion of fish that we caught that were around Gobi was almost 50%. By 2007, 2008, these are lumped together so I could get all the data on one. But yeah, by the very next year, you're looking at 75% or greater, almost a monoculture of fish, right? So we just have dominant round Gobi. And that's it. That's before, oopsie, before and after. It's crazy. So that's a lot of stress on our native fish, because if round Gobi is making up 75 to 90% of the biomass of these little forager fish that we catch, then they're also eating that much of the food, and they're taking up that much of the habitat and breeding grounds and whatnot. And so it's not, it's a great example of what happens when an invasive species comes in and how the other organisms respond. And and so that's a story that we're seeing, but it's not the only story. That's the story that we're catching. There are other threats to our native fish caused by climate change that we are seeing anecdotally as we're moving into. We don't record this in our sales, but we see this as an organization. The last 2 years, our bay has not iced over until March. This year, it never fully iced over. And there was only ice in our little docking area, like the very tip of the embayment, for less than a week. When you don't have ice cover, you increase your wind, your turbidity, your water clarity goes down because sediment and guck is stirred up. And if that's stirring up your eggs, right? If you're laying eggs in those shallow zones, they're no longer protected by the ice. Your habitat's being eroded. You're getting, there's a lot of negatives associated with a lack of ice cover. Less ice fishing, I suppose. A positive for our native fish is that there are more spring food sources, because food runoff and things is carrying food sources in a little earlier. So they are getting access to food. So the fish that make it into the spring have more food sources, and that can be really good for early spring survival. So one thing that we're digging into in our data, but haven't yet had the time to extrapolate, is, are we seeing more fish caught in our little bay now than we have? Are those fish, are we seeing a higher number? Even if we're not seeing a lot of diversity, are we seeing more than we have seen? That's something that we're exploring. And we are also obviously seeing warmer temperatures. We see that in the data we collect throughout the sailing season, and we see it off season, although we're not recording it then. We're seeing more and stronger storms that result in less oxygen in the summer. The same fish that thrive in early spring then have to swim far deeper into the offshore. So are we catching less fish later in the summer as the water heats up? Because that habitat's no longer conducive for them. And that also triggers some weird signals for spawning and other biologic functions. And all of these things are caused by climate change, but because of our 32-year record of fish catch, we can go in and explore what the impact of these things are. So this is an example from whitefish. There's some tribal biologists here in the region who are also looking at this. They've put out a bunch of tools to track where the whitefish are going in the throughout the seasons, and then correlating those to catches. And what they think happens is they have to swim off further offshore in the summer in winter, especially the larger fish populations, to find food and to stay comfortable temperature-wise in the summer. And the big impact isn't to the adults, but to these young fish, right? The ice cover is protecting those eggs and larvae. And when the ice cover isn't there, it's a lot harder for the young to survive to adulthood, and then for the adults to find appropriate spawning ground when the time comes. And this is an example of what researchers I think are happening with the whitefish, but the same thing is being seen on lake trout. There's some research that shows that some lake trout are staying in place, and then their signals are getting skewed, and the growing is retarding. So older fish are far smaller than you would expect for their age, because they're not going into deeper waters. They're choosing to they're being really flexible in staying in that warmer water, but it's making them less healthy as an organism. So those are really interesting things that we're looking into, and you need a longitudinal data set to do. This is a cool example of just a lot of the connections between the food web that we collect data on every day and some of those bigger systems, things like climate change. So if I move us again here to somewhere else, cover the round Gobi. Quagga and zebra mussels, they're increasing the water clarity by decreasing the plankton, but they're also providing nutrients to encourage cladophora growth. Cladophora growth becomes abundant. It's the stuff that grows on the bottom, and it loosens and it dies, and then it creates biomass. It gets all stirred up in storms, and then it creates biomass that is then dead and then sinks to the bottom and rots. When it rots, the things that eat the rotting material eat the oxygen as well, and they can cause hypoxic or anoxic conditions. And then you can also even create the botulism toxin. Fish can eat that decaying material. They can get botulism. The mussels are consuming it, the fish are consuming it, then these larger consumers like ducks are consuming them. They wash up on the shore, and then animals are consuming them. It's a really cool and really complex diagram that just shows you how many unintended consequences a couple of small shifts can really have. And the only way you can see some of those things is through those longitudinal data sets, which I say all of that to just say every single time somebody gets out on our ship or any research vessel and does collect with us, you're allowing us to paint that picture, to tell the story for one more season, for one more year, and help us understand these big processes, because we all know the only constant is change. These are some of the cool people that I've been gifted enough to work with. I just want to point out that so many of the people who are educators and scientists with us are actually volunteers who are giving of their time. And we invite you to join our community by bringing your kids out on the ship, or visiting us in the summer, or coming to a teacher educator PD if you are ever interested. We would really invite you to join us and hang out with us. If you ever want to, there's our information. And I really thank you guys for joining me and letting me talk a little bit about some of the cool ways that collecting fish and plankton and scoops of mud with kids has led us to a broader understanding of what's happening in Suttons Bay and throughout the Great Lakes. Thanks.
Thank you, Trish. Thank you for bringing this incredibly rich material. I just cannot imagine how much you gave us just within this past 40 minutes. So thank you so much for this great presentation. I remember hanging with you guys last summer with two of my students. when we were working on the Climate See Soon documentary, and we are very, very excited to be able to share it with all of you already in a few weeks on April 6th. So, we enjoyed so much being with you on the boat and learning from you and being a part of your group. Could you tell us a little bit more about how other educators can either engage, with the experience on the boat, or perhaps just with the data sets? If if we are far away, if we cannot necessarily be passengers on your boat, how we can engage with data?
Sure. So first, I think shooting me an email or signing up. I shared the PowerPoint presentation, so I'm happy for anybody to have a copy of that, or shoot me an email and I will connect you to the right person. Right now, our data sets are a bit in a bit of a transition phase. We have them. There is a lot of data, and so we are working with Field Scope to actually make that data digestible and accessible for teachers. And one of the things we hope to have, I don't want to say by the end of the year, I hate to, tell you we have this cool data set and you can ask those kinds of questions, and then say, but you got to wait a little bit, but you do have to wait just a little bit if you want an easy solution, because Field Scope, eventually there'll be a quick link right on our website. You'll be able to click it and it'll give you a data output. So you can say, I wonder what fish have been doing since from the day I was born to the day, like kids like to ask that question, what has it done for the last 8 years? Or what was the Secchi depth the year I was born? And so that is coming in the near term. Right now, we are happy to connect you to that data, but it comes with a much bigger Excel CVS file. Some of it is in Access, so there are some database software skills necessary, but we are willing to work and share that data with anybody who is interested. And we are working on, we have some, I think they are fairly old. They were taken off the website, but if you reached out, I could connect you to some supports for that data if you were interested in exploring with your students or exploring yourself. And all of our fish data is also, we share it with Eagle or DNR. We legally are required to have the permit for fishing that we do. And we also have, we have those Otter Trail locations, the latitude and longitude coordinates, and we put out like minnow traps, and so we can tell you what we catch if we just put out a minnow trap and how that's changed too. So we do have a lot of really interesting data. It's just not quite in a digestible format now.
Thank you. And I remember that you had many students, both undergraduate and graduate students, interning for the inland seas. Was it just like something ad hoc, or you regularly have those opportunities? I see we have a number of students on this call in the audience, and I'm sure they will be very interested to learn how to engage with you. Yeah,
we have college students to thank for a lot. And so thanks for tuning in. I would say that we have had a few different master's level projects come out of the database work, working to systematize our processes, sort of from like a logistical, how can you do citizen science in a way that is the data is replicable enough to share out, and then how do you find the right platform to make it digestible for students. We've had folks actively using our data sets for their research. We have a PhD student right now out of, I believe, Wisconsin, who when we catch sticklebacks, we preserve them and then ship them to him. So because he just needs more, he's doing some genetic work, I think. And so we just ship him the fish. And then they send us their research. When they're done, they send us the papers they've written. And so we are lucky enough to be a part of a lot of those projects. The University of Michigan Biostation, they have a undergrad, grad level field course that they use us, and I think some at least abstracts have come out of. So I can thank college students for forwarding our knowledge, using our data to forward the collective understanding in really important ways. We unfortunately spend unfortunately, maybe not right. We spend a lot of time out on the ship teaching, which means we don't get to play in the data as much as we would like. To get involved yourself, in addition to if you're doing that level research and we can be helpful in any way, please email us and we will do what we can. Our program survives on volunteer and internship educators. We are a small but mighty staff. We just hired our first person whose job is to educate last week. We all have other jobs, so having someone whose time is dedicated to being an educator, to being out on the ship, frees us up to do those other jobs. But because of that, interns are on almost every sail that we do from May to October, and they grow themselves as educators. They grow their understanding of the Great Lakes, and so wherever their path takes them, we really appreciate it. I don't know if we still have internship slots available, but you could check our website. I know we've been interviewing a lot the last week or so, so I don't know if we filled everything yet, but so there's always internships available. We have housing that comes with that, as Elena mentioned, so we can house folks. And we have different lengths of internships, be it 7 weeks, 14 weeks, and you get to go out sailing with us. And so that's a great way to be involved. We also have a number of folks who volunteer either as educators or as crew because they're curious about sailing and just want exposure to the Great Lakes. And sailors are some of the best meteorologists that I know because they have to integrate that atmospheric knowledge for safety. And so you learn an awful lot about science by just being out in the elements and connecting with nature in this way. So those are all ways that you could get involved, and we'd love to see you.
Thank you, Trisha. But to remind everyone, please send your questions through the Q&A. I have a couple of questions here already. Dr. Bidande is asking, very interesting, do you expect the mussel Gobi dominance to continue for many more years?
Yeah, I think with mussels, probably yes. I know there's some researchers doing some cool work, basically putting glorified tarps over tracks to kill off the mussel, and that's having some success. So I think they might be targeting specific areas to do that. But I think the positives on that, the dracenids have become part of the food web. And so gobies and dracenids came from the same area over in the Caspian Sea region. And so they already were part of a food web. So gobies eat them, but only up to a certain size because gobies are pretty tiny. But we're finding them in perch, we're finding them in lake trout now. So the native species have started incorporating the mussels into their diet, which is really great for the recovery of those native species. So I think they're here now. And then as far as gobies go, the research, the most recent work I've read is probably out of date now. But they look for, I guess, a common way to explore how established a population of fish is looking at the ratio of male to female in a population. And we've actually incorporated that into our high school level program to start getting male and female gobies. So we're sexing them now. And what you can expect, they're very aggressive. So they tend to, like, their younger males tend to really spread out quickly. And so the number of males to females, the ratio is higher in an actively in a population still growing and expanding and dominating, and it becomes closer to 1-to-1 or 2-to-1 male to female when they become more established and sort of part of the ecosystem. And in most of the Great Lakes states, it's a 6-to-1 male to female ratio. So I think we can expect the gobies to continue to do their thing for a while yet. I don't know if Janet has any other information on that.
No, I think that was very well said. Actually, we have another question from Dr. Bananda, and he's inquiring where you got the Seki disc earring.
It's a great question. My colleague is amazing. She likes to be the person people get Christmas gifts from. So these were Christmas gifts that she made for all of our staff.
Great. Love it. Oops.
Thank you very much. Thank you.
Yeah, thank you
Janet,
guys.
have any additional questions or comments? Well, thank you, Trisha. Thank you very much for sharing your wisdom with us. Lots of wonderful information. We hope to see you soon again. And we start running out of time, so it's time to say goodbye, unfortunately. It's so great to see you again.
Thanks, friends.
Yeah. Thank you so much. Bye.
Thank you.